Hi, my name is Alex Ray. I work at the Nanomaterials Research Group here at Western, and I'll be talking about the effects of doping bismuth Boro-Tellurite glasses with rare earth ions. So what can you expect today? First, I'll tell you about why we do this work, give you a little bit of information about glasses and what it means to be a glass. I'll show you how we prepare our samples, and then I'll talk to you a little bit about each of the different measurements that I make on these glass samples and what that means to us. So why do we do this? One, rare earth ions like praseodymium and dysprosium, the one I'm using in these glass samples, are known to have these high outputs of fluorescence, and those can be used for different types of lasers or color displays or other types of LEDs that can be used in different industries. And then the doping of glasses with rare earth ions is interesting for other sorts of industries like solar cells or photovoltaics, who are also interested in research of doping rare earth ions into materials. So hopefully our research can do something to help that type of research as well. So why do we choose the different components that we use in our glass? We take bismuth oxide, borate, tellurium oxide, and we put those together as kind of our host glass. The borate's really good at making a glass at varying different percentages for composition. So we use that borate kind of as our base, and then tellurium oxide and bismuth oxide have high refractive indexes and they lower phonon energy, which gives us a little bit more energy available for our fluorescence. And then the tellurite or the tellurium oxide will reduce the melting temperature for us. If you're interested in learning a little bit more, there are a couple papers that the research group has put out recently that talks a little bit more about it. So then also we're using praseodymium and dysprosium as our rare earth ions because of the very strong emissions they have at blue and red for praseodymium and blue and yellow for dysprosium. So what makes a glass? Usually whenever a liquid cools down very quickly, it does so and makes a crystal. So if you look at the figure here on the right, it will go from the liquid, this melt, down to a crystal, and it loses enthalpy very fast. So that means that one, it's losing a lot of heat, but two, it's also creating a structure that is periodic in nature. So it's very organized. For our glasses, they have this glass transformation behavior where instead of getting very organized into these long periodic structures, they're a lot more disorganized so that they have different structures in different parts of the glass. So we get these local environments that we're interested in that can affect the fluorescence. So we get this amorphous structure. It's not the same all the way through the glass. So we prepare these samples here at Western ourselves. If you look here in the bottom left, I have one type of sample that has 10% tellurium oxide, and then we change the dysprosium content from 0.5 to 1 to 1.5. And then we also have a second type of sample, which has 20% tellurium oxide. And I do the same thing for that, where I change the dysprosium content. So we can compare dysprosium content change as well as comparing the difference in 10 to 20% tellurium oxide. So I take these compositions, I weigh the amounts, I mix them in a mortar and pestle, I put them in a crucible, then we melt them at about 930 degrees Celsius. That melt is done very quickly so that we don't lose any of our volatile tellurium oxide. And then we quench them on these brass plates that have been preheated to about 100 degrees Celsius, so that the temperature doesn't drop down too quickly. Afterwards, after the quench, we have these samples that are fairly fragile, and they break really easily, and we put them in an annealer right after that. So the annealer is going to raise the temperature to about 325 degrees Celsius. This is close enough to the melting point so that the molecules inside the glass can move a little bit, and they can kind of release the thermal stress that's within the glass, making it so fragile. So the annealer brings it close enough to the melting temperature that it won't melt, but it can release this stress, and then it slowly brings it back down to room temperature so that the product after the annealer is a lot stronger We can then use this lap machine here to smooth the sides so we get nice flat sides and polish it. So it looks like this finished sample here on the right, where it's see-through and each side is flat. The first thing that we measure after making these samples is density. We do so by using Archimedes' principle. Our samples aren't in a nice geometric shape that we can just, measure the radius and the thickness and all that and get a sort of density that way. So we have to use Archimedes' principle. So using some quick algebra, we can get this equation here where we just have to find the mass of our sample in air and then the mass of our sample in a liquid and the density of that liquid. We use xylene so that it won't react with our sample. We know the density of xylene, so we can use that to find the density of our object. So we use this setup here that is homemade. We put our sample here in this little orange basket. We can take the mass in air, fill the beaker here with xylene, and then we take the mass in xylene, and then we can quickly calculate our density. When we do so, you'll see for the 10% tellurium samples here, you'll have a decrease in density as we increase the dysprosium content, which is interesting because the dysprosium is a heavier molecule than the borate that it's replacing. So as we increase dysprosium, we're decreasing borate slightly. So we're increasing the mass. So when you increase the mass, you would think that the density would increase as well. But since it's not, we can say that the structures around the dysprosium, the structure within the glass, are creating more volume to accommodate for this extra mass. So bismuth oxide, tellurium oxide, and borate all make their own structures within the glass, and how they fit together is being changed by this addition of dysprosium. So that those local environments are changing slightly, and it's reflected in our measurements here. So for 20% samples, we're seeing a similar thing here at first, where the density is decreasing, but then it increases later, which is also kind of interesting, which means something with these structures must be fitting together a little bit better there at the end, with our 1.5% dysprosium content that has the 20% samples. So this point here is saying that maybe the structures are fitting together a little bit better, but also if we look at this as a whole, the density isn't changing very much. We have about somewhere between 5.80 and 5.85 here, 5.75 here, and then 5.85 here. So this isn't changing all that much. So it could not be super significant yet. We'll have to look into that a little bit more, but right now you see a kind of interesting increase. The next thing we look at is refractive index. We do that by measuring Brewster's angle. Brewster's angle happens when P polarized light is reflected off of the surface. You get a minimum light reflection. So whenever there's the least amount of P polarized reflected, that's your Brewster's angle. And we can use Brewster's angle to find our refractive index using this equation, where N1 here is just the refractive index of air, which is 1. So this is our setup where we take this laser, it goes through a polarizer, which gives us half S polarized light, half P polarized light, goes through our slit, hits our sample, reflects off our sample, and then we focus it onto the photodetector here. The photodetector will tell us the intensity of the light that's being reflected off of our sample. We use our analyzer here to kind of, or to get rid of all the S polarized light. It filters it out so that we only get P polarized light on the photodetector, and that way we can find the minimum. So we change the angle of the photodetector by 1 degree around what we think should be Brewster's angle, and then when we get closer to what we've seen in other types of glasses, we start going by 0.5%. So if you look at this, we get a nice quadratic here, and we can get the equation of that quadratic by analyzing the data. We then minimize this quadratic equation to find our Brewster's angle down here like this, and then we use that Brewster's angle to find our refractive index. So again, if we look at 10% samples for refractive index, we're getting a decrease as we increase dysprosium content. And as a general, refractive index and density usually correlate pretty well. They don't have a direct relationship, but they do have this correlation usually. So we can see that it's decreasing. So again, this is due to some sort of structural changes. If we look at our 20% samples, again, the refractive index is about the same for each of our dysprosium content change. So as we go from 0.5 to 1 to 1.5, those are about the same value. And that, again, that's similar to what it was for our density measurements. So everything with density and refractive index is telling us that, one, they're green, and two, there are some structural changes occurring within the glass. The last thing that I measured was fluorescence. Fluorescence occurs when there's some sort of incident light that hits our sample, and then it will excite the electrons in the sample. Those electrons, when they go back to the ground level, will emit photons. So this requires a specific amount, or specific amount of energy in order to excite those electrons. So for praseodymium, it's 445. For dysprosium, it's 355 and 458. So we're using a 445 nanometer laser, which is only going to excite the praseodymium. And then we look at what is our fluorescence after we excite the praseodymium. So our setup goes like this. We have our laser light, 445 nanometers, reflects off of our sample, hits this filter here. The filter takes out anything from 450 nanometers or below, so that our laser isn't going to be into our spectrometer. We then focus that light into the spectrometer. The spectrometer bounces the light off of some mirrors and through a grating so that it separates all of the different wavelengths. Our photomultiplier tube increases the intensity of these detected wavelengths, and then our computer reads them. So this is what it looks like whenever our spectrometer gives us an output. You can see for our 10% samples here on the left, we have this one peak that's increasing as we increase dysprosium content. And same thing for the 20% samples. This is increasing as we're increasing the dysprosium content. So there is something considerable happening that we need to be looking at here. So for our 10% samples, after we analyze these this fluorescence data, we can see that the peak that's increasing is for 574 nanometers. And this big peak is for 602 nanometers. So the interesting part is that this peak for 574 nanometers is for dysprosium. So it is one of the dysprosium electronic transitions, not praseodymium, even though we're only exciting the praseodymium. And if we look at our 20% samples, we get the same thing, this peak at 574 nanometers again. So what's happening here? Well, first I compare these peaks, or I take the dysprosium peak, the 574 nanometers, the area under that peak, and then compare it to the ones for praseodymium, and I get this so that we can see there is a clear increase going here on how much of, or how big that praseodymium peak is. So then what's happening? Dysprosium is increasing. This dysprosium peak is increasing, but we're not actually exciting dysprosium with our laser. So what we can say is that since we're only exciting this praseodymium, as the praseodymium is fluorescing, it is somehow exciting our dysprosium. So we're getting this cross excitation between praseodymium and dysprosium that we're not doing directly. So within the glass, the praseodymium is exciting this dysprosium, which is an interesting occurrence. So to conclude, what are we seeing with our classes? Number one, we're seeing that there are some various obvious structural changes occurring. Our density and refractive index graphs show that, and some further measurements could probably tell us a little bit more about what these structural changes are. So we can do Raman spectroscopy or IR spectroscopy to kind of figure those out. And then we're seeing this cross excitation with praseodymium and dysprosium, which is very interesting. And again, we can look into that a little bit more, maybe using some absorbance spectrums. And then if we want to maybe increase our fluorescence after we learn a little bit more about it, we can maybe try some new variations of dysprosium content or other types of content in order to kind of get more out of this. So this is where I want to acknowledge WIU, my supervisor, Dr. Muller, and the research collaborator, Dr. Bebu, for all the help and letting me do this research. Thank you.